Okay, so now we're going to watch a video of a eukaryotic cell dividing, just so you're able to see all of the steps of cell division carried out as a whole. So this right here is the eukaryotic cell, and inside of it is the chromatin, which is just a decondensed version of chromosomes. And right now the cell would be in interphase, so it's really just going out throughout its regular activity, and the cell is growing and the DNA is replicating and it's starting to prepare for division. So you wouldn't be able to see these steps exactly under a microscope. So now what you're going to see is the chromosomes started, or the chromatin condensed into chromosomes. So now they're more visible to see. They have the bar-like shape. So the cell would be considered to be in prophase now because the chromatin condensed into chromosomes, and the spindle is starting to form in this step as well, which you won't really be able to see either, but you can tell that the chromatin definitely condensed. So now the cell would be in prometaphase, which is where the nuclear membrane did dissolve, which you can't really see from this video, but you can see that the chromosomes are starting to move, and they're moving with the aid of what's referred to as microtubules, which are pulling the chromosomes. They're starting to pull them to the center of the cell. And then in the next step, which is metaphase, what you'll see is that they're completely going to align in the centromere of the cell, which is just the center. So here you can see they've lined up right across the centromere, or what they refer to as the metaphase plate in the cell. So now the cell is going to move to anaphase. This is where the chromosomes, which are lined up in the middle, are now pulled apart and separated into what are called chromatids, and they're pulled to opposite poles of the cell by the spindle that was formed in prophase. So now the cell is going to go ahead and move to telophase. And here, this is the what's going to become the nuclei of the two daughter cells. So now the cell is moved into telophase. So the chromatids decondensed back into chromatin, and the cell is starting to separate into two separate cells. So as you can see here, it's starting to pinch in right at the centromere. So that would be telophase. And now cytokinesis is going to complete the rest of the process by completely pinching off this center part here, and you're going to have two separate daughter cells, so the cell will have completely divided.